Please, a round of applause as we welcome Pro-Chancellor, sir. Welcome, sir. Professor M.C. Asuzu. You're welcome, sir. Professor in charge of the Office of Management and Strategy of the university, Professor Adeduro Adegeye. You're welcome, sir. Permit me also to warmly welcome and recognize the second inaugural lecturer of the university. He delivered the lecture just a month ago, his inaugural lecture, Professor Leroy Edozien, a professor of health, law, and policy. I will also likely to welcome the first inaugural lecturer of this university, the person who delivered the first inaugural lecture of the university is here present, Professor M. Simidile Odimayo. Thank you very much. I have the privilege to also warmly recognize and welcome on this occasion Father in the field of food science, Professor Isaac Oluwalana. A round of applause for Prof. You're welcome, sir.
She lectured at the University of Ibadan from 1985 to 2018. During that period, she rose from the position of national lecturer and a Pavia PhD student of Roseangela who newly founded Federal Pathology Program. She has led several research projects, both as co-principal investigator and principal investigator. Today, it is a pleasure to ask Prof. to take the stage as the give a lecture.
Good morning, everybody. The Vice Chancellor, Deputy Vice Chancellor, Administration and Clinical Services, the Registrar, Librarian, Deans of the faculties, distinguished ladies and gentlemen. It is a pleasure and honor to deliver this third in the series of inaugural on behalf of the Faculty of Science of this great university. My heart is full of praise and thanksgiving to the Almighty God for granting me this opportunity. I could never have imagined that my coming to this university from the University of Ibadan, after being accepted by the pioneer Vice Chancellor Friday Okonofa for just a one-year sabbatical leave in 2015, will turn out this way. I was first, when I came here, appointed the pioneer of the Department of Biological Sciences, and now Deputy Vice Chancellor Academic. It can only be God. He has done so much for me, and I cannot turn it off. This lecture is the first to be delivered from the Department of Biological Sciences, which was recently split into two departments, the Department of Microbiology and the Department of Bioscience and Biotechnology. And it's also the first from the Faculty of Science. The previous inaugural lecturers from the university were Professor Michael Odemayo, with the title, Turning the Tides Against Infectious Diseases. And the second, titled, My Body is My Property, by Professor Leroy Edozian. In my years graduates, I found myself studying zoology in the University of Jos. And that was the year that JAMB started a general matriculation admission board in 1978. So I found myself in zoology. I say it's an unguided choice because I just chose zoology to pick up with what I wanted to do for my undergraduate. But at that time, I was an A-level student in the Federal School of Arts and Science, Aba. And the rule was that you don't submit to admissions for JAMB. And we were the first set. So I decided to split and choose different courses that are far away from each other. And I ended up not getting admission for my A-level and ended up getting the admission for the entrance exam, which was zoology in UniJos. So that was the journey I found myself in Jos, out of the situation, and moved into the world of cell biology and molecular parasitology during my final year. So I found myself in the university, and I decided to choose some courses that are more or less chemistry inclined, because I loved chemistry and I wanted to read biochemistry or industrial chemistry. Interested in the biochemical reactions and activities of parasites, and that was, I keyed into it. And he accepted me as his undergraduate student, and I did a project which was the nematode parasite, characterization of the nematode parasites of that, really in line with parasitic nematodes and trying to identify their functional Krebs cycle enzymes. So when I graduated from UniJOS, as a zoology major with the best results in the department, my dad was insisting that I do a postgraduate course because he didn't see zoology as a specialized course. He was an engineer. So to go for my master's, and I chose the University of Ibadan in the Department of Zoology. And I think it was an act of God, because normally everybody expected me to go back to Jos. But I decided to go to Ibadan after praying about it. So in Ibadan, I felt a little bit lost because it was a new environment. But there was a culture in the department then where postgraduate students interacted with their lecturers, most of the professors and the heads of department. And the interaction get us talking. We decided they would ask you what you're interested in and so on. And I found out that Professor Nwuba, who became my mentor, was interested in studying parasites of trypanosomes, and he was interested in the glycolytic enzymes of trypin on something like that. I have a project, and right now I don't have any master's student. Will you be willing to work with me? And it was like the miracle of my whole life, because that's what I wanted to do. I wanted to work with enzymes, which is more chemistry inclined, and he was there offering me this opportunity. So during that period, I did my master's. Although I came in for parasitology, I did my master's under the cell biology and genetics unit, which is very rare. And I had to beg Professor Ukoli, who was the sole lecturer in parasitology, to allow me to do my project under Professor Nwuba. He reluctantly agreed, and I thank God for that. Because when I look at all that happened, it reminds me of the quotation of King Solomon in Ecclesiastes 9:11, where he said, I return under the sun, that the race is not for the swift, nor the battle to the strong, neither yet bread to the wise, nor yet riches to men of understanding, nor yet favor to men of skill, but time and chance happeneth to them all. In my case, the chance happened to me for good. And according to Professor Nwuba's narrative, that was the same time in his research career he was contemplating of setting up a molecular parasitology program within the department. So everything just worked well. I was the first student to show interest as a PhD student or master student, which later graduated to my PhD, being the first PhD student in cellular parasitology. So cellular parasitology was coined out from these ideas of looking at the parasites in the molecular and cellular aspects. Professor Nwuba had been in the lab of Christian de Duve, who was a Nobel laureate in 2017 in physiology of medicine, and he worked on glycosomes and peroxisomes. And he got him to apply for a grant, which he succeeded, and that started the landmark of working with trypanosomes. And I was there willing to work with him. Slide 5. So I finished my master's program under Professor Nwuba, and the idea then was that cellular parasitology was built on the premise of the idea of the doof. And he created all the sophisticated ideas, environment, equipment for me to do my work at that level. He was working on the regulation of glycosomes in trypanosoma brucei, looking at two enzymes, hexokinase and cellular parasitology. The main objective of that cellular parasitology program was to equip young scientists in parasitic diseases prevalent in Nigeria and Africa, to the end that cellular parasitology program attracted people from all over Africa, from Kenya, Cameroon, Gabon, and they were all there to study the cellular aspects and molecular aspects of parasites. So I'll deviate a little to the issue. What are parasites? What are parasites? Parasitology can be defined as the study of parasites, and parasites are simply organisms that live inside or on another living organism called the host. And they survive by taking nutrients from the host. In other words, they live off other organisms to survive. But parasites refer to protozoans, which consist of only one cell, to worms like helminths, which are larger and consist of many cells, and they have internal organs. And also to ectoparasites, which are mostly insects and can cause and transmit diseases. So these are the groups of parasites as I just described. So the title of my topic today is actually beating parasites at their game. So what type of games do these parasites play? That's mostly parasitic disease are mostly linked to poverty, which are mainly endemic in tropical Africa. Mr. Vice Chancellor, sir, in the situation where the hosts in questions are human beings, it's evident that research scientists like us, that you need to know your enemy and know yourself also before you can fight 1,000 battles without disaster. So in knowing your enemy, you need to study these parasites. Study their cellular regulations and everything, and that's what we are on doing in cellular parasitology. So my research work was mostly focused on winning the war on disease. Disease and cancer is real, and we find ourselves have talked about this. So next, nature has provided an explicit natural strategy of anticipating what will happen to the host. So we develop immune systems to fight the parasites. And this immune system can be either innate or adaptive. So all this sophistication are counteracted by the parasite by developing defense mechanism against our immune system. So one way strategy that parasite take, strategies that man develop against parasites. These strategies can be qualified as strategies of interception, anticipation, intervention, and silent adjustment. And the parasite themselves try to counter these quotes of Professor Nwuba that says that development in areas of basic biology has now deeply transformed the prospects of contemporary medicine, since it's no longer held back by the impassable boundaries of cellular world. So medical research can now gain full entrance into the cells, and it allows itself to be guided by modern and cellular and molecular biology. So that was the main reason and the main stand of our research. So my PhD started with a project called the glycosomal phosphofructokinase from T. brucei. So I was working on African trypanosomiasis. So this trypanosomes has a complicated life cycle, and within the life cycle, it has a unique organelle called the glycosomes. And the glycosomes have all the enzyme, the key enzyme, phosphofructokinase. We found a paper was published from Trinity College, Dublin, where my project that was supposed to be carried out for the next 3 years was being published, and that's a scientific nightmare. So the publication came up, and Professor Nwuba saw it and said, wow, Roseangela, we are in real trouble. Your project has been published, so you can't even do much about it. And I'd gone far trying to purify the enzymes. So when we got this news, we said, okay, let's do some other aspect that they didn't do. Only for 6 months later, for another publication from the same source. And at that point, we knew that we were really in real trouble. So Professor Nwuba decided that, you see, you must go to this lab and complete your PhD. Whatever that's left out, you continue with them. So he applied for WHO to give me a training scholarship to go to Trinity College to complete my PhD. And that was what I did. On getting to the lab, Kit Tipton, who was the supervisor, gave me the lowdown of what have been done, what is left to be done, and that is what I specialized and did my PhD on. Work was mostly looking at the effect of pH on this enzyme. They had done the substrate concentration and every other thing, so I was just looking at how pH regulation affects these enzymes. Rotation into the glycosomes. You see, the glycosome itself was a membrane-bound organelle in trypanosomes, and this enzyme was found within the trypanosomes. I would say God turned everything around for good, because in so doing, I got the best training in enzyme kinetics, a specialization that my supervisor then had little in, and I finished my experimental aspect of my PhD, which attempted to, like I say, look at the kinetic effects of pH on this enzyme. And see, the life cycle of trypanosome is quite complicated, and it infects mostly the few parts in South Americans. So the rationale for my PhD project was to selectively destroy a parasite within the host by exploiting unique differences between their metabolic pathway without incurring substantial structural damage on the host. There was a shift, and this major shift was because we got a huge grant to study malaria. And you all know in science, who pays the piper dictates the tune. So we had to change our mode of research. That came around to fund MIMTIDER was in Europe, founders in Europe, USA, and all the other ones, with the aim of bringing the best out of scientists in Africa. The laparositology program benefited with a project called Antibodies, Malaria Merozoite Surface Protein 1, Processing and Erythrocyte Invasion. Merozoite Surface Protein 1 is an important membrane protein of the trypanosomes. Actually, the red blood cell red blood stages of the parasite. And this is the aspect of the parasite that produces fever, the symptoms of malaria. So we looked at this antibody. We are now looking at an option of enhancing human immune system with the goal of contributing to vaccine development rather than the first focus of fighting the parasite. The antibodies produced by merozoite surface protein 1 include a processing inhibitory antibody, which stops the parasite from invading red blood cells. So that's the merozoite on the other end. And before the merozoite can invade the red blood cell, it needs to undergo proteolytic catalysis reaction. And if it doesn't, it cannot invade the red blood cell, which goes with the parasite into the red blood cells. If you can stop that inhibition, the parasite doesn't invade the red blood cell. It's reason to study this parasite. Before then, studies have shown that in vitro, the parasite can produce such inhibitory antibodies. So this is the same, not a simple, it's a complex assay, but you can see that if you have antibodies that stops inhibition, you will not see a proteolytic reaction between MSP-142 to produce MSP-133 and 14. But most of them also produces proteolytic reaction. That means these individuals do not produce processing inhibitory antibodies. So this is what the processing inhibitory antibodies does. Processing from going through, the whole MSP1 remains intact and nothing happens. Invasion does not go. But if invasion occurs, the processing inhibitory and the parasite will invade the red blood cell. Another interesting aspect is that the parasite itself is also playing another game with us. In as much as we can produce processing inhibitory antibody, we found that the parasites also, when exposed to a red blood cell, to the immune system, produces blocking antibodies. These blocking antibodies will stop the processing inhibitory antibodies from preventing invasion. So it's like a competition. You can see this green bad guy here. That's the processing inhibitory and the blocking antibody. It blocks the processing inhibitory antibody from binding and allows invasion to take place. So this is the type of war we are facing, game we are playing with these parasites. As to try to fight them, they look for another way of fighting back and brings us back to square one. In fact, most of our studies show that in humans, we have even 3 types of antibodies. The ones that block, the ones that inhibit, and the ones that are neutral, they are not doing anything. So if you use the antibody titers to find out whether you're functioning, your immunity is functional, you'll be deceived because you produce all these antibodies that don't do anything. Mostly, they are either blocking to prevent the real antibodies from functioning, or they're just neutral, doing nothing. So our project moved also into a follow-up to characterize the dynamics of antibodies in malaria in humans exposed to malaria parasites. And this design had the goal of critically now looking at the information of the functional characteristics with age, because we don't know whether these antibodies are produced mostly in young children or in adults. And we did a cross-sectional study in Ebola to find out. But before then, Professor Nwuba had concluded his work, and the results we showed was very encouraging. And he was about retiring from the university. And the WHO recommended him highly for training young scientists like myself, Dr. Nwuba, Professor Daibo, within the department that could carry on this work, even in his absence. So I wrote a proposal, and I got this proposal. I became the PI for this proposal, for this work. Mr. Vice Chancellor, sir, my training had given me the opportunity to stand on my own, apply for grants, and win grants. So you can see that they studied that just by a mere chance of working with Professor Nwuba brought me to a stage where I became a PI in my own department and became the head of cellular parasitology program when he left. So some of the results we got in our subsequent projects was pointed out to the fact that these processing inhibitory antibodies were even age-related. By the time we looked for processing inhibitory antibodies in adults, there was little or no processing inhibitory antibodies in adults. But we still found the three specific types, the neutral, the blocking, and processing inhibitory. But the level of these processing inhibitory antibodies were very, very different according to age. So between the age group of 5 and 12 years old, we had the highest proportion of children producing processing inhibitory antibody. None was found below 6 months, and in adults, we had just one adult out of a very large group study that produced processing inhibitory antibodies. So we were wondering now, what's the game this parasite is playing? We found out that mostly non-specific antibodies were being produced, or the blocking or the bad ones. And since the antibodies were not really relevant, that's why we still come down with malaria. So we try to see what is giving, we know that immunity increases with age. So what is actually confining this immunity to man, if it's not the processing inhibitory antibodies, and MSP1 specific, moreover. So we looked at what are the MSP1 specific antibodies that the adults have that inhibit processing, and they are not MSP1 processing inhibitory antibodies. And we were lucky because we brought a breakthrough where we had a genetically modified MSP1 that could recognize antibodies that stops evasion, and you could recognize whether it's MSP1 specific. And we can also use another test to know whether they are processing inhibitory antibodies. Mr. Vice Chancellor, sir, although the human immune system evolved a strategy to beat malaria parasites on its game, I think the body also started developing means of conquering all these blocking antibodies by developing their own type of antibody that can stop evasion and yet cannot be blocked by the blocking antibodies. It's like an icing on the cake of the pinnacle of my career. I've done a lot along the line in the University of Medical Sciences before coming here. And coming into UNIMED, first it was so like a barren land because it was just starting. The labs were not as equipped as the ones I was used to in UI. But I just felt it was just a one-year thing. I'll just come and go. But even within that 1 year euphoria, we started looking for how to get grants to equip some of our laboratories since we are not getting funds from the state government. So we started looking for grants. We got the Seeding Labs grant that Seeding Labs just gives you instruments, equipment. It's an instrumental grant that equips your lab. If you have an idea of any research and you write to them, they will look at it and competitively weigh it with other applications and give you a grant so that you can equip your lab and start your research. Fortunately, we applied for Student Labs and we got that for University of Medical Science when I was the head of department of Biological Sciences. So we got that grant to equip our lab so that people can do research. I came from a culture of labs that can do research, win grants, and encourage and build capacity. So this is what my UNIMED story was mostly all about. So apart from that, we also won an NRF grant from TED Fund, where we were looking at the studies of dizygotic twinning in Ebola. And remember, most of my research work were done in Ebola, although it wasn't on the twinning. But in doing research in Ebola, I noticed that twin births in Ebola was very rampant. And Ebola is actually has the highest twinning rate in the whole world. So it's curious, like, why are they producing twins? Is it their diet? Is it genetic? What is it that is having Ebola concentrating, a small town like Ebola having the highest twinning rate? So, and the former Vice Chancellor, Professor Konofa, was also interested, even before I came here, in twinning in Ebola because of the reproductive angle. He wanted to know if the twins were due to diet and which diet, so that we can exploit it for fertility. If we have diets that, so why go and buy fertility drugs? So it's coincided that both of us and Professor Mokwa also, that was a twin, came up to write a proposal. And this proposal won an NRF grant to look at the twinning rate in Ebola. But not just the twinning rate. Also, if genetics has a role to play, and not just genetics, whether there is a link between the environment and the genes, which is the epigenetics. So epigenetic studies are looking at the way the environment, which includes the diet, whatever you eat, relates with, interacts with the gene to produce something different. It doesn't change your genes, but it changes the way the genes are expressed. So the expression of the gene will differ depending on the environment you find yourself. So this search, we won this grant. We've written some papers on the results. The epigenetic studies stalled a little bit because of the COVID-19 lockdown, and also because we couldn't get the reagents from foreign companies that had just stopped producing anything. We are concentrating on more of COVID-19 testing because that's where the money is. But the arrival, we've got a test kit, and we've done that, and we've ordered for more kits to complete this research work. And with public work on the idea perception of the people from Ebola, why they have twins. Some said it's just an act of God, and so on. Some said, pointed it to diet. And we are going to be looking at whether diet also affects that. So, there's also an aspect of building capacity in molecular bioscience and genomics, which we've extended to this university. It's noteworthy that our team also have developed 3 molecular biology-based laboratories. I told you about the seeding labs, and during COVID-19, with the help of the Vice Chancellor, the state also built and equipped a COVID-19 testing lab, and also the TED Fund also built and equipped a second molecular biology lab, which is on campus. So we have a full-fledged molecular biology lab with well-equipped laboratory that has been NCDC certified, that can carry out any top-notch research. It has a 24/7 power supply located at the General Hospital here in Ondo. So we can see that by the results of the game playing, I will pose this question. Is beating malaria parasite at its game a mirage? You can see that each time man comes up with something, even in chemotherapy, they develop resistance to drugs. You come up with vaccines. That's the one aspect that is the parasite finds this difficult to beat, because the vaccines are targeted directly to the parasite. In October last year, the WHO rolled out for the first time, malaria vaccine for humans. It's called the RTSS vaccine. And it's from the protein that surrounds the sporozoites. It's called the second sporozoite protein, and they fused it with hepatitis B to increase the antigenicity of the body to produce more of this protein. Significant feasibility or statement percent of people that have been given this vaccine. For my friends and beloved brethren, I cannot say a lot, but to say God bless you. Special thanks to Professor Olumide Ogundaunsi again, and everyone that has contributed for the success of this event. I also acknowledge my family members. Some of them are here, and some are not here. I'm grateful for my late parents, Engineer and Mrs. Moloku, that nurtured me and brought me up to this stage in my life. His Royal Highness, my uncle Igwe Achebe, the Obi of Onitsha, I'm also indebted to you for your role you continue to play in my life. For my darling husband, Engineer Mark Nwuba, the crown upon my head, you have been there with me. You've been there with me, and I give God all the glory for choosing you as a life partner. My lovely three children, Chiamaka, Kosi, and Toby, you are simply God's precious gift to me. Ladies and gentlemen, thank you, and God bless you all for coming.
The university anthem.
For research that we've get to all to achieve the best in health sciences, build a nation, change the world, touching lives and making impact in the nation is our goal. Touching lives and making impact is how we will grow. UNIMED. make an impact is how we will grow. UNIMED, UNIMED, giving all for a better tomorrow. UNIMED, UNIMED, that's our help for a greater nation.
Professor of Molecular Parasitology. Okay, today's lecture is looking at the ways scientists like me in cell biology and molecular parasitology can explore various avenues to beat the parasite at its game. I started my research work with trypanosomes, which is a parasite that causes sleeping sickness. And these parasites are protozoans, and they have a unique organelle. It's a unicellular organism. It has a unique organelle called the glycosomes. Because they are catalytic enzymes and for energy production that keeps them alive, it's embedded in the glycosome, which is an organelle that is unique to that family. It's laid upon our heart to study this enzyme in the parasite, exploit the differences between the mechanism of this enzyme in the parasite and in man, so that if we can develop a drug to attack that enzyme, it will not affect man because their mechanisms are so different. So that's why the study of that enzyme. It's a very key regulative enzyme. Blocking it or inhibiting that enzyme will cause the death of the parasite. So this is one major aspect of my work. But after that, I moved into studying malaria parasites. The malaria parasite is also a protozoan parasite that causes malaria. It's called Plasmodium species, and the Plasmodium parasite has a life cycle in human, which is the main host, and mosquitoes. But I wasn't interested in the aspect of the mosquitoes, but the aspect of when the mosquito bites you and introduces the parasite in the body, how do we stop it from reproducing, from dividing, developing into a disease? So if you can halt it at any point in time, we chose the aspect of the life cycle that invades the red blood cells, because most of the fever and chills and headaches we get during malaria is because a parasite had invaded the red blood cells and multiplied within the red blood cells to form merozoites. These merozoites are now released. These merozoites are now released in the bloodstream. So your body reacts to it because it sees it as foreign and starts trying to attack, develop antibodies to fight it. That's why you have this fever and you're shivering. It's the way your body is trying to evade or react to the foreign organism within our system. So that's the aspect we were interested in. So we wanted to see how we can study what the parasite does, the immune system does when it sees the parasite, and see if we can boost it with a vaccine that will increase the immunity of man. That's what vaccines do. Vaccines mimic the parasite, enable you to fight the parasite when the parasite is not even there. But once the parasite has arrived, your body has recognized that parasite before it will be able to fight it better. So, in studying the immune system, we had to look at what happens in human and what happens in the lab. Most of the time, foreigners culture parasites in the lab, do their experiments, come to conclusions, but they don't suffer from malaria. So we are the ones suffering from malaria. So we want to know what the aspect of the things they saw in the lab, is it really what happens in nature? So we had to do a field study, collect blood samples, check what type of immunity we are developing against malaria, the type of antibody, classifying it. Do they have the same type that they find in vitro, in vivo? So for the first time we found out that actually humans produce processing inhibitory antibodies. What does this processing inhibitory antibodies do? The processing inhibitory antibodies are the ones that stop the parasite from entering the red blood cells. So if they stop that and it works, that means you won't suffer from malaria. Professor Roseangela.